Hello, my name is Jared McHugh, and this is my presentation on the urea cycle. In this presentation, I plan to outline the purpose, function, other facts, as well as the biochemical aspects of the urea cycle. First, we're going to get into the overall goal of the urea cycle. The overall goal of the urea cycle is to manage the levels of liberated ammonia present in the tissues of the human body due to protein breakdown. The urea cycle was the first metabolic cycle to be observed in the human body in what could be considered a photo finish. The urea cycle was discovered in a matter of days before the aptly named Krebs cycle by Krebs and Henseleit in 1932. And yes, I mean that Krebs. The person who discovered this cycle later had the citric acid cycle named after him. And for anyone unfortunate enough to become very acquainted with the Krebs cycle, you should be very familiar with that name. The urea cycle is a cyclic reaction that removes excess ammonia from the bloodstream and converts it into water-soluble urea, which can be effectively excreted through the action of the urinary system within the human body. And it is a mechanism of nitrogen balance as well within the human body. Okay, now let's get into where the urea cycle occurs. The urea cycle occurs in the liver, in particular the mitochondria of the liver for the first two steps and the cytosol within the native tissues of the liver for the last three steps. Interesting fact about the liver is that it has one of the highest concentration of mitochondria within its cells, which gives it a relatively high capacity for facilitating this series of reactions. The products of the urea system, while not necessarily relevant to the process besides its yield, are processed through the urinary system. Due to the water-soluble nature of urea, it is a very easy metabolite for the kidneys to properly process, package, and pass out of the body. The nephrons, aka the functional unit of the kidney, is the site of removal of the urea from the bloodstream through glomerular filtration. Alright, now let's dig into the why of the urea cycle. Ammonium ions are a common byproduct of the process of proteolysis within the human body. In an unbalanced amount, ammonium is toxic and leads to a multitude of issues within the human body. The most prolific of these issues is present in the nervous system. Excess ammonium interrupts potassium transport in the brain, leading to impairments in its functions. It is examples like this that renders the urea cycle an absolute necessity with regards to homeostasis within the human body. Okay, so let's talk about the steps that lead us up to the urea cycle. As I mentioned before, proteolysis, which is the structural breakdown of proteins within the body, occurs throughout the body for a wide variety of reasons. Proteins, of course, occupy many roles throughout the body. Structural use, enzyme formation, fluid balance, and general body function leads to a turnover rate within the human body around 1 to 3% per day of its total yield of protein. The breakdown of these proteins leads to an increase in nitrogenous waste within the body. This is where transamination comes into play. Transamination is the transfer of an amine group to another molecule. In the human body, the most common molecule responsible for this is alpha-ketoglutarate, which bonds with the liberated nitrogenous waste to form glutamate. Glutamate is able to flow freely within the circulatory system, which makes its way to the liver to be further processed. Continuing on, glutamate is responsible for safely transporting the bound ammonia to the liver without allowing ammonia to freely flow in the bloodstream, which would lead to issues regarding toxicity. Glutamate, once in the liver, undergoes a process called oxidative deamination, which is the breakdown of glutamate into alpha-ketoglutarate and a liberated ammonium ion, which now resides in the tissues of the liver. Alright, now we're getting into the real nitty-gritty of the urea cycle. Liberated ammonia, present after oxidative deamination, condenses with carbon dioxide that is present after phosphorylation of bicarbonate. These molecules condense with the help of two molecules of ATP and carbonyl phosphate synthetase 1, which ligands these molecules together to form carbonyl phosphate and two molecules of ADP, or adenosine diphosphate. This portion of the reaction occurs in the mitochondria within the cells of the liver. Now into step 2, the formation of citrulline. The second step of the reaction occurs when carbonyl phosphate undergoes a transfer of its carbomoyl group into ornithine, which yields a molecule of the amino acid citrulline. This reaction is facilitated by the enzyme ornithine transcarbamylase. Ornithine is peculiar in that it is non-proteinogenic and essentially only plays a role in the urea cycle, hence why this cycle is commonly also referred to as the ornithine cycle. The third step in this cycle takes place when citrulline, with the help of ATP and catalytic action from argininosuccinate synthetase, to form a molecule of citrulyl AMP intermediate. This molecule condenses with aspartate to form argininosuccinate. This is also the first step of the reaction which takes place in the cytosol, due to the expulsion of citrulline from the mitochondria in the previous step. Something else to note, this is also the only point in the reaction where ATP is converted to AMP due to reactions taking place within the urea cycle. The fourth step of this reaction begins the systematic breakdown of argininosuccinate. Like step 3, this part of the cycle takes place in the cytosol within the native tissues of the liver. This step takes place when argininosuccinate lyase catalyzes the breakdown of argininosuccinate into the molecules arginine and fumarate. Here we come to the final step of the urea cycle. This final step also occurs within the cytosol in the native tissues of the liver, and it occurs based on the hydrolysis of arginine. Arginine is hydrolyzed, which leaves us with the products of urea, our final product, and ornithine, once again making an appearance in this cycle, giving note to its cyclic nature. This process is catalyzed by the enzyme arginase, and also requires the presence of water. Urea, being a polar molecule, is very soluble in water, as I mentioned earlier on in this presentation. This solubility allows it to be easily excreted by the urinary system within the human body, thus, by nature, properly preventing the buildup of toxic levels of ammonia in the human body. Coming full circle, here is a brief summary of the urea cycle. Overall, the urea cycle is an effective way to manage the waste produced by amino acid catabolism and is necessary within the human body for the purpose of homeostasis. In short, ammonia is converted to water-soluble urea. Nitrogenous compounds are always contained. This prevents perfusion of toxic ammonia into the bloodstream as well as the surrounding tissues of the liver. In this system, nitrogen is always bound until it is safely excreted out of the human body via the human urinary system. On this slide, I wanted to make note of some of the enzyme activity and how it relates to clinical practice. As we can see, enzymatic activity is prevalent in this cycle. As we see across the steps, we see 5 individual enzymes responsible for catalyzing the steps all along the urea cycle. However, given the nature of enzyme production in the human body, it is not completely unheard of, however, for there to be deficiencies in enzyme levels, which can lead to what is termed as urea cycle disorders. Disorders of the urea system can lead to a variety of issues that, generally speaking, will need some form of professional clinical intervention to manage. Disorders of the urea cycle generally lead to an excess buildup of ammonia within the cycle, which is termed as hyperammonemia. And as we can see in a study of Tahonen et al. 2010, interruption or production of the balance of enzymes is generally what's going to lead to what is professionally and clinically termed as a urea cycle disorder. Some further interesting facts regarding urea cycle disorders. They generally speaking can be managed long-term with clinical intervention. Generally speaking, there are lifestyle and pharmacological treatments that can be implemented to prevent long-term issues associated with urea cycle disorders. The most common of which these disorders carry are hyperammonemia, neurodevelopmental deterioration, and unfortunately weight gain. These conditions are usually diagnosed in childhood due to the high rate of presentation of the associated symptoms, but can actually manifest in some patients after pregnancy. Common minimally invasive treatment options include implementing a low protein diet and either a form of sodium or glycerol phenylbutyrate. These medications, interestingly enough, are actually salts of aromatic fatty acids that provide an alternative route for urea to be excreted from the liver. And as someone who works in a pharmacy, I do actually see these types of medications come through relatively common. So I found this to be a bit of an interesting fact to connect with this project on the urea cycle. And now a dip into the ever evolving world of transplant medicine. I found this personally interesting. It is a little bit down the pipeline is some severely, I shouldn't say severely, but incredibly advanced clinical practice. As once again, someone who works in a pharmacy, I see a variety of different transplant management plans, treatment plans, and it is an endlessly fascinating aspect of medicine. Interestingly enough, there is mounting evidence that shows that liver transplantation is a potential intervention to prevent many of the associated long term effects of urea cycle disorders. The double edged sword bit with this, given the fact that there isn't exactly a surplus of spare livers floating around, this more than likely won't become an extremely common treatment for quite a while. However, hopefully, due to advances in medical technology, we could potentially look forward to treatment methods based around this theory in the future. And thus concludes my presentation on the urea cycle. I hope you enjoyed listening to my little take on the urea cycle, as well as some of the interesting aspects of how it applies to human life, human homeostatic function, and some potential interesting future clinical practices and applications of the urea cycle. Personally, I found this project very interesting as it gave me a way to connect concepts of biochemistry into my intended school path, in my current job field as working in a pharmacy. So overall, I was very excited to take on this project and see what I can garner from it. And I cannot wait to see all the different ways that this is going to pop up in my future. Thank you.